starting our today's session with the assertion and reason based question. Let's look in the assertion. It is saying the A base substitution in DNA. So the modification or the mutation is the base substitution in the genome level in the DNA doesn't always result in mutant phenotype. So the mutation is happening in the genotype and you are not able to see the mutation in phenotype. Is it possible or not? So it is saying about the base substitution mutation. There is another mutation that is called the indel mutation. That means insertion or deletion. So the insertion, so a nucleotide from the outside that could be inserted in the open editing frame, or a existing nucleotide from the open editing frame that could be deleted. So this kind of mutation will totally change the frame of the codons. The codon frames, if a single nucleotide is like deleting or getting inside the ORF, the open editing frame, that means the total frame, the total codon frame will be changed. Now, so there will be like the protein that will be produced, there will be no desired amino acids. Every amino acid, every single amino acids will be changed in that protein. Thus, it will make a lethal protein. And if the insertion or deletion mutation is happening, well, I have told about the indel mutation for a single nucleotide. And if it is happening for 3 nucleotides, then the severity of the mutation could be less. So like here, that is one codon, another codon, another codon, and another codon. And if this codon is getting mutated or another codon is coming in, like either it is going out or another unknown codon, the 3 nucleotide is coming in, and it is not harming, if it is not harming the frame of the codons, then the level, the severity of the mutation might be less than that of the single nucleotide insertion or deletion. So it is not saying about the like indel mutation, it is on the base substitution mutation. So before going to that, we should know the like a special feature of codons. Like we know the codons are degenerative in nature. That means a single nucleotide can be produced from more than one codon sequence. Like there are 64 codons, and among them 3 codons are stop codons. So 61 codons are there, and from these 61 codons you will get 20 amino acids. That means for a single amino acid more than one codon is there. That is called the degenerative nature of the codon. And here comes the Ubel hypothesis. It is saying if that is a codon, so if that is a codon 5 prime to 3 prime, so this will be the anticodon. This will be the 5 prime to 3 prime. So the binding between this for the codon for this first 2 nucleotides will be more specific than that of the third nucleotide of the codon and the first nucleotide of the anticodon. So this is the anticodon sequence and this is the codon sequence. It is in the tRNA. This anticodon sequence is present in the tRNA and this codon sequence is present in the mRNA. So the first from the codon side, the first 2 nucleotides will be having the specific binding, but the last one that might have or might not go for a very much specific binding. From this picture you also can see this, like here in the case of proline, like just see all are having the third nucleotide. The third nucleotide, only the change is in the third nucleotide. First and second nucleotide is same. And also in the case of threonine, the same thing you will be getting. The alanine, the same thing you will get. The valine, the same thing you will get. Also for the glycine, arginine, and in lot of cases you will get the serine. You will be able to see this kind of activity. So the Ubel hypothesis might be the reason behind this. So if you are changing on the Ubel position, on the third position, like from the picture also it is visible, like the nucleotide was, the codon was UCU, and now you have changed it to UCC. Still, it will produce the same amino acid, that will be the serine. That means there is a mutation in the genotype level, but in the phenotype level, the mutation is not visible. So the assertion A is possible. So the reason is saying that some base substitution in codons do not change the encoded amino acids, that we already have seen. So both are right. So both A and R are correct, and R is the correct explanation of A. Let's move on to the next question. One is regarding the polycystic mRNA that is found in the archaea and bacteria. So what is the polycystic? What is the polycystic RNA? So rather being a type of mRNA, we should say it is a special kind of conformation. The structure of mRNA. A series of genes is present in a polycystic mRNA. So in the that is only present in the archaea and bacteria. That is true. That means it is only present in a prokaryotic system. So that is the specific structure, like a, that is the promoter binding region, and thereafter operator might be there, and thereafter you will get the series of genes. So this kind of conformation is known as the polycystic mRNA. So under the presence of a single promoter region, so more than one gene is present, more than one mRNA is present. That only happened in the case of prokaryotic system. But in the eukaryotic system, under the expression of a single promoter, you will get only a single mRNA. So that is the just that is the conformational change in the case of like prokaryotic system. And what is the utility of being of formation of this kind of polycystic mRNA in the case of prokaryotic but not in the case of eukaryotic? So eukaryotic system, there you will get a lot of kind of, that is a very much complex thing, and like the alternative splicing and other kind of things are there. So that is not present in the case of the prokaryotic system. And they also have to maintain a very short length of genetic material. Their structure is very much short size, and their genomic structure is also very compact. So to make it, to give the genome the compactness, so more than one mRNA should be expressed under a single promoter. If we are making different system, like in the monosystemic, in the that is happening in the case of uh, eukaryotics, then it will be very much difficult to maintain their whole genome. And now this kind of structure gives them another kind of like advantage. So now, like if that is the lac operon system, like the operon systems are having this specific kind of structure. Like in the operon system, there is the another portion, and there is another promoter. So under the expression of this promoter, the repressor protein will be produced, and it will come and bind on the operator region. Thus, it can control the expression of the proteins, the expression of the mRNAs, those are present under a single promoter. And now, if you look, so all the genes that those are present under the that series, so that those are the different genes, different mRNAs sequence. So their function is somehow related. Like in the case of LAGZ, so there you will get the production of the like beta galactosidase enzyme and lactose permease enzyme. So those kind of enzymes activity is related. If the lactose permease is not activating, if the lactose permease is not at all present in the cell, so the lactose will not be able to go inside, and there will be no work of the beta galactosidase enzyme. So thus the conformation happens in that way. So this polycystinic mRNA construction is very much important to decide the and regulate the operon system. And one more thing, if all the genes that is present in a bacterial system or in the archaeal system, if they all are active, that might make it the lethal. That might be a cause of their death. Because if every gene is expressing, if there is no lactose, you need not to produce any kind of beta galactosidase or galactoside permease enzymes. So if you are producing it, that means you are wasting your energy. And in the case of bacteria or archaea, if they are wasting their energy behind some production of unwanted proteins, that might be lethal for them. So that's why this kind of structure is too much important. And now it comes the statement 2. Statement 1 is right, and statement 2, it is saying about the plastid gene in eukaryotes have only monocysteinic mRNA. Although the plastid is a organelle inside the eukaryotic system, but they are not having the only monocysteinic RNA is not present there. They are also having some kind of polycysteinic mRNAs. That comes, there is a theory of endosymbiotic, like those are like uh, endosymbiotic, like bacteria, or like here you can say the certain organelles within a eukaryotic cell that is originated from from ancient bacteria which was engulfed by the cell, and thereafter they form a symbiotic relationship, and over time they evolved into organelles. This kind of organelle is the plastid. So endosymbiogenesis is the theory behind the formation of the plastid genes, or also you can see in the that plastid is present in plant cell, and for animal cell the same thing happens for like mitochondria. So how the people know about this? So depending on their specific kind of structure, like both are double membrane bound organelle, and they can produce certain till a certain extent they can produce their own proteins, and also they are having some polycystinic mRNA structures. That gives the scientists the idea that they were not in a association with the eukaryotic system when they were originated. Thereafter, after some time, there was the relation, very strong correlation, and thereafter the eukaryotic system has like engulfed their plastid plastid organelles. So nowadays that is the organelle, but that time it was the free living bacteria, and most probably that mitochondria is from alpha proteobacteria and plastid was from a cyanobacteria, and thus the evolution happens, that is known as the endosymbiotic theory. And so the statement 3 is right. Human genome contains approximately 25,000 genes, but the number of protein in a human cell is estimated to be approximately 250,000, that is 10x of the estimated number of genes. So, what could be the reason behind this? So, first of all, the alternative splicing will be the major contributory in that case. So, what is the alternative splicing? Like, if this is your genome sequence and the red portions are showing like intron sequence, and blues are the exon sequence. So, that is the exon 1, exon 2, and exon 3. So, itself, they can make three separate genes, but when they will go for the splicing, for the alternative splicing, the magic will happen. Like 1 and 2, they can form another gene. 2 and 3 can form another gene. 1 and 3 can form another gene. And also, they all together can form another one. It is 1, 2, and 3. So, from 3 genes, you are getting 3 single genes and 3 double genes and 1 like triple genes in together. So a single gene will form here, here and here, and now 2 genes together will form a protein, and here all together will get, you will get another protein. So from 2 genes you are getting, here you are getting 3, from 3 genes you are getting 3, 3 and 1, 7 kind of 7 genes. That is the beauty of the alternative splicing, that is exclusively present in a complex eukaryotic genome. And another thing is the post-translational modification that is also present in a eukaryotic system, not the protein folding. So what is the protein? What is the post-translation modification? Post-translation means after the production of the protein. So, so in a eukaryotic system, you can see some isoforms, the protein isoforms, and lot of kind of modifications like ubiquitination, like phosphorylation, acetylation, and the glycosylation. We have already seen that the glycosylation is very much crucial for its, for the activity of the chaperone inside the rough endoplasmic reticulum cytosol. And like the receptor tyrosine kinase, the phosphorylation is very much important to make the protein, to make the receptor in a active form, to give it a active form. And also in the case of histone modification, so the phosphorylation, the acetylation, like, and methylation, this kind of things are very much crucial to regulate the activity of the histone. And the modified histone will give the specific structure, will control the euchromatin or heterochromatin structure, the, like the expression of the target genes. That thus it will make the epigenetic regulation. So the post-translational modification is too much important to regulate the activity of the protein. Once the protein was in inactive form, after the modification, it might go to the active form, or it might show some another activity. Thus, post-translational modification and alternative splicing contributes in this kind of situation to, from this, like they are increasing the fold by 10x. Only 25,000 genes are able to make 250,000 estimated human proteins. So the fourth option is the right one. And here is another thing, is the post-translational silencing. So silencing can happen from miRNA. If it is indigenous, then the miRNA, and for experimental purpose, you can induce the siRNA. But this transcription, the post-transcription, means in the mRNA level, the modification is happening in the mRNA level, that is silencing. That means you are silencing a specific gene, a specific mRNA. That means the diversity will go down in that case. So the fourth one is the right answer. Telomerase enzyme has the following activity. We have to determine which one. The activity of the which one resembles the activity of the telomerase enzyme. So from the picture that is on the right side, we'll see the activity of telomerase enzyme. So before that, we have to know where there is a need of this kind of enzyme. So telomerase, from the name itself, you can get it is a polymerase. So the polymerization is its activity. So now what is the need of this telomerase enzyme? So when there is a replication, like if you are taking this one as a template strand and this is 3 prime and this is 5 prime, so here you will get a RNA primer, and thereafter the DNA polymerase will bind on the RNA primer and it will extend to synthesis the daughter strand, and thereafter so the synthesized DNA will be like this, where this is the 3 prime to 5 prime and this is 5 prime 3 prime. Now this is a RNA, this is a RNA portion, RNA primer. It can't stay in a mature DNA. So soon after the synthesis, the completion of the double stranded DNA synthesis, this portion is going to be removed. So now there is a 3 prime overhang of the DNA, of the mature DNA, and so this portion is the 5 prime. So the DNA polymerase can't extend that way because it can go 5 prime to 3 prime. It is having the 5 prime to 3 prime polymerization activity, but 3 prime to 5 prime it can't like extend. So there would be a hollow, and from this another problem will arise. So this portion, this portion is will stay in a single stranded format. So now if there is any DNase enzyme present inside the cell, then it can come and cut the DNA that is present in a single stranded form, and thus we will lose the genetic information that is present at this position. And from the each cycle of replication, the same thing will happen. For that, after certain cycles of DNA replication, the whole genomic information will be lost, and thus the cell will die and the genetic information will also be deleted. So if that is the situation, then the surviving for a organism is not possible. Here comes the activity of the telomerase enzyme. So now that is the problem I told you. So this kind of DNA you are having after the replication, and now the telomerase enzyme will come, and this is a ribonucleoprotein. It is having a like inbuilt RNA sequence. So this one here is the inbuilt RNA sequence, and like depending on the sequence complementarity, like here this is the sequence complementarity. It is from the overhanging region they will find the sequence complementarity, and after making the complementary region, the rest region will be working as a primer, and the DNA will be synthesized on that way, and now the DNA is in a extended portion. So here the primer can come and bind. Now the primer can come and bind. In the first case here, the binding region of the primer was not there, but now after extension of the 3 prime overhang, the primer will get the sufficient place to come and sit. So this portion is for the from the primer side, and thereafter depending on the activity of like DNA polymerase, it will be extended on that way, and thereafter again the primer will be removed. Now there again will be a hollow, and so that is the first cycle of the activity of the telomerase enzyme. So again the telomerase enzyme will come, and this cycle will continue for certain period of time. And in this case, the like the telomerase is providing the template. So it is having a RNA template. So it is depending on the sequence complementarity, it is giving the the sequence that is having the 3' overhang, it is giving the primer, and thereafter it is also having a activity that is the RNA dependent DNA polymerase activity. That means depending on this RNA primer, it can synthesis the DNA. So here this is the RNA primer, and the telomerase is having the RNA dependent DNA polymerase activity. That's why it can extend this 3' overhang by introducing the DNA nucleotides. Thus the telomerase works. Now, where should the telomerase stay? The telomerase is generally it is present in high amount in the stem cells. But if it is present in a differentiated cells, that will be very much difficult condition. Like in the differentiated cells, they are having a certain kind of lifespan. If the cells are having a certain lifespan, so and you are just coming and repairing again and again the DNA molecule, that means the cell will remain for a long period of time. So that what is the how that if the cell is differentiated, then how it is going to be destroyed or going to attain the apoptosis. So in the differentiated cell, you will not get the telomerase. That's why it will start losing the genetic information, and after a certain period of time, that will trigger the apoptosis and the cell will die. But that thing should not happen in the case of stem cells. That's why this kind of polymerase, this telomerase, should stay in the stem cells only. And if it is increasing in the case of differentiated cells, that means the cancerous condition may appear. There is a cancer that is the melanoma, that is known as a type of skin cancer. In that case, it has been observed that the telomerase activity is higher in the cancerous cells. And like if there is a if the activity of the telomerase goes down in the in that in the cells, that is the stem cells, that means in that case some complexities like the progeria, the disease progeria, can happen. That means the aging before the actual age. So that was all information about the telomerase enzyme. So you need this all to secure a good marks and to understand, at least to have a more understanding on the topic. So now the correct answer is the RNA dependent polymerase. This telomerase activity is very much important, and regarding this topic, you can expect some questions in your paper. So let's move on to the next one. So we have reached the end of our today's video, and this is gonna be the last question for today's session. This is a assertion and reason based question. So that is a very much basic and simple question. You also can answer this question very easily, but I kept this question in the slide because from this you will have a you can get a knowledge that this kind of questions will also be in your exam paper. And from the texture, you might think this is a tough question. So if you are opting for this, you might lose your time. But this question is actually very easy. And so this kind of questions will also be there. Just don't forget about this and try to grab and manage your time. Try to grab the marks and also manage your time. So you have to select the perfect, the easy questions. Let's come to the question. So the strand separation of DNA occurs in vivo during processes such as DNA replication and transcription. So in the transcription and DNA replication, if it is happening in vivo, the strand will be separated. It is whether it is happening in vivo or ex vivo, in both the cases the strand should be separated. So now in the DNA replication case, the DNA will be like separated in two single strand form, and thereafter the primer will come, and that will the DNA polymerase will come, and the polymerization will be happening, and thus the DNA replication needs a single stranded form. After the replication, the newly synthesized DNA will have a strand from the mother, and one will be the daughter strand. That means the DNA replication is happening in a semi-conservative model. And next that is the transcription. In the transcription, like a bubble should form on the initiation site where the RNA polymerase can go and bind to its specific location, its target location. So in the case of transcription, the two strands you will get. One strand will be the sense DNA strand, and another one will be the antisense strand. And the mRNA will be synthesized from the antisense strand, and it will be having an exact copy of the sense strand. But the T will be replaced with U only. So the first option is correct. And the reason is the sites where these processes are initiated are often rich in C and G base pairs. So in the in vivo processes, the helicase enzyme will be involved to melt the hydrogen bonds that is present in between the double stranded DNA. From the double stranded DNA to produce the single stranded DNA, you have to melt the hydrogen bond that is present in between two strands. So the helicase enzyme is responsible to do that, and if the initiation site is rich with C and G, that means the number of hydrogen bonds are high in number. So it will be very much difficult for it to break down. Rather, if it contains the high amount of A and T sequence, it will be very much easy for them to break the lower amount of hydrogen bonds. Thus, this option stands wrong. It should be A and T. You might have heard about the Tata ad box. So those kind of box are there on the initiation site of the replication. So from there it is clear like you need a rich A and T sequence. So the A is correct but R is not correct. So far we have gone through 5 questions, and I tried to make it easy so that you can internalize all the kind of questions. And if you are having any kind of doubt or any kind of issues, you also can tell us, and you can write in the comment box. And for now, till that, so goodbye, and hope we will be meeting in the next video. And stay happy.